The topic is reproduction in plants. What is reproduction? Reproduction is one of the basic activities of the living organisms. All living organisms have so many vital activities in their life. Reproduction is one of the important features by which the organism perpetuates its own progeny to the next generation. Whether reproduction helps the plants or animals or any organism to perpetuate its own species, whether the function is over with that, no. By the time when reproduction process is on, gametogenesis takes place. During gametogenesis, reduction division is taking place, which involves the crossing over. Crossing over is a process in which the genetic materials are interchanged between the chromosome, as a result of which recombination of chromosome is produced that results in the variations of species. So, reproduction helps for perpetuate its own species. Not only that, it results in variation. Whether variation is required, yes, variation is essential tool for the organism to survive in the future generation or to survive in the changing environment. And this variation results in the evolution of species. Results in evolution. So, reproduction is one of the important features of all living organisms. How many kinds of reproductions are there? There are two types of reproduction. One is asexual reproduction and another is sexual reproduction. What is asexual reproduction? Asexual reproduction is a method by which an organism produces or perpetuates its own species without the involvement of gametes. So, in asexual reproduction, no gametes are involving. No gametes are involving in the asexual reproduction. So, what are the methods in asexual reproduction? We will see one by one. In asexual reproduction, in lower organisms, lower plants, animals, etc., microorganisms, etc., they produce certain special asexual structures like conidia, gemma. Then the method called fragmentation, and budding, regeneration, and binary fission. These are the methods in lower plants and animals adopted by the organism to reproduce asexually. Conidia. See, conidia are produced mostly in fungi, lower plants and fungi. Fungi is also coming under this. In fungi like penicillium and aspergillus. They produce conidia, a special structure through which they reproduce. These are asexual spores. For example, we are drawing the aspergillus, the structure of conidia and aspergillus. These are sterile hyphae upon which conidia are produced in all directions. Conidia spores. These are the spores, conidia. And gemma. Gemma cups are produced. This is a method of asexual reproduction adopted in Markensia. You know, Markensia is coming under bryophytes. Markensia in the thallus, if this is the thallus, a cup-shaped structure near the median ventral groove is produced, this can be detached. In due course, this is detached from the plant and falls on the suitable substratum and produces a new plant. And fragmentation. Fragmentation is most probably seen in these Markensia also, but especially in Spirogyra, algae like Spirogyra. Algae like Spirogyra, the filament, if this is the filament and cells, and there is a fragmentation in certain places, and the filament is detached. One filament becomes two filament or three, like that. Every filament, the cells in every filament divide and produce a full filament, so that from a filament many filaments are produced. This is a kind of asexual reproduction in Spirogyra. And budding. Budding is found in yeast. It's a method by which yeast reproduces asexually. If this is a cell of yeast, it produces a young yeast in one place, budding. And also in the animal like hydra, you can see new hydra are produced by the process called budding. So these are the examples for the organisms which reproduce asexually through budding. Then regeneration. Regeneration means producing the lost parts and getting to the natural state. So this is found in the worm planaria. Planaria. See, you would see the structure of planaria like this. If by accident or by the unexpected application of mechanical force over that, the planaria is cut into two segments, and every segment, the upper segment will regenerate the lower segment, and the lower segment will regenerate the upper segment, and two planaria will be produced. This is regenerating, producing the lost part again. This is called regeneration. This is found in planaria. The next is binary fission. Binary fission is found in bacteria. So this is nothing but the division of the cell, the vegetative cell, into two entities by a simple division is called binary fission. See, if this is a bacterial cell and an invagination on both sides happens, that grows towards the center, that make the single entity into two parts, and each one will develop into the organism. This is binary fission. So these are the 6 methods. You see, 1, 2, 3, 6 methods of asexual reproduction by which lower organisms or lower plants, fungi and animals reproduce. And because of this, they produce clones. Here, what is the speciality? See, if an organism reproduces by means of asexual method, the progeny produced will be having 100% similarity with the parent plant. So what are clones? Clones are the younger organism or generations of organisms which are produced asexually, which are similar in morphological and genetical characters. So this is a group of assemblage of organisms which are similar with one another morphologically and genetically. They are from the same parental source, and these are called clones. So asexual reproduction produces clones. Clones are the organisms which are morphologically, genetically similar with each other, and this is over. this is found in asexual, this is found in lower organism. What happens in higher organisms? Vegetative reproduction. In higher plants. See, vegetative reproduction in a lower plant is carried out by 6 methods, 6 type of structures or methods. And vegetative reproduction in higher plant is carried out by 2 methods. One is natural method. Natural methods. In natural method, what is natural method? If there is natural, there should be artificial or other things. We will talk about that afterwards. What is natural method? Plants propagates asexually by their vegetative organs like root, stem and leaf. What is the meaning of that? Part of the root, part of the stem or part of the leaf or any advantageous structures produced in the root, stem, leaf can be utilized for propagation. So this is a kind of adaptation found in higher plants. So usually the root, stem and leaf of higher plants are meant for perennation. And also storage. But in some cases, especially the root, stem, leaf produce special structures or advantageous buds that will involve in the propagation of new generation. So these structures we are involving in the propagation of new structures, they are called diaspores or vegetative propagules. The root, any structure of the root, stem and leaf is involving in propagation of the next generation, propagation of the plant. They are named as diaspores or vegetative propagules. So in natural methods, root, stem, leaf are involving in they themselves without any external stimuli. They involve in the propagation. They are called natural vegetative propagules or natural diaspores. And we will see which I said root involves in vegetative propagation, stem involved in vegetative propagation. This is vegetative reproduction. We are talking about vegetative reproduction. What is vegetative reproduction? Vegetative reproduction is a method of asexual reproduction by which the organism reproduce or propagate with the vegetative parts like root, stem, leaf. I think you understood this. And we know that they are meant for perennation and storage. If they involve in that, they are named like this. Now we will see vegetative reproduction in leaves and stem and leaf one by one. So vegetative reproduction in roots. Vegetative reproduction in roots. See, vegetative reproduction in roots. Say my example, Mureya, Kunuhi, Kuniki, Mureya, Kunihi, is a best example for that. See the root, then Millingtonia and Dalbergia, plants are also involving in this type of propagation. See, you would have seen the Murraya plant, Murraya curry leaf, Murraya plant, if the stem is there and here are roots, and some roots or aerial, they produce an advantageous bud that can grow into a new plant. So this type of reproduction is found in Millingtonia and also in Dalbergia also. This is a kind of vegetative reproduction in which root actively participate in the production of new generation, next generation, or perpetuation of the next species. So actually this is not next species, it's own species with the own genome. So this is a kind of reproduction. And next is stem. Second is vegetative reproduction in stem. This is a bigger topic where we have to discuss the different aerial and subaerial stem modifications and their involvement in the asexual reproduction. See the stem, part of a stem or advantageous buds produced by the stem or involving in vegetative propagation. Actually, you should remember the advantageous buds are involving in the propagation. In stem, they are all modifications. There are subaerial modifications and underground modification. In subaerial modification, there are 4 types. There are 4 types. One is runner, second is stolen, third is offset. Runner is Centralia. Example for runner is Centralia asiatica. Here the stem grows horizontally on the surface of the soil, and at every node it produces leaves, Centralia asiatica. If by the application of mechanical force, if it is cut down or the stem is cut, and this can grow independently, this is a kind of vegetative reproduction which is found in Centralia asiatica, and there are so many examples for that. Then stolen, it is found in Fragaria and Mentha. Fragaria and Mentha, it's the same type, but the only one difference between the runner and stolen is the horizontal stem bends upward a little and touches the ground, and here you can see the leaves are produced in clusters and advantageous roots. These are all advantageous roots, and leaves are produced in cluster. So this is a difference between this is a tuft of leaf and these are advantageous roots, and here if there is a cut made, and each one will develop into a new plant. And offset, so offset is found in Eichernia and also Pistia. So this is a water plant where the same runner, the type of stem is found with tough leaf over upper side and cluster of roots hanging in the water. This is water surface. The offset, the difference is the distance between two successive nodes is little less, and so it is called offset. This is found in Pistia, the water plant. And fourth is sucker. Example, chrysanthemum. In chrysanthemum, what is special? Here there is a main trunk which has two parts. One is aerial part of the main trunk and subaerial part of the main trunk. So some part of the stem is also buried inside the soil. From the axillary buds of the subaerial part, lateral branches are produced and they grow upwards obliquely, producing crown of leaf. Crown of leaves like this. There will be advantageous roots over that. And if this is detached, this can grow into an individual plant. So these are the four types of subaerial stem modification in which the axillary bud, actually axillary buds are involving in the propagation. Axillary bud found in this and also here, involving in the generation of new species when accidental breakage or mechanical force is applied unexpectedly over the plant. That can be detached and they can grow independently over any place if the substratum is good for its growth. And next is underground stem modification. We will talk about underground stem modification. As we saw in subaerial stem modification, the underground stem modification, there are four types. One is the rhizome. Second is corm. Third is tuber. Fourth is bulb. We'll see one by rhizome. Example, ginger, turmeric, and banana, for example, for rhizome. See what happens actually in this. See, in rhizome, is the underground stem grow horizontally in the surface of the soil. So we think that this is a ginger. Here are these lines represent the node. In between two nodes there is internode, and in the place of node there are certain structures like this. These are called axillary buds. I said in the earlier time that axillary buds are involving in this asexual reproductions, and this grows horizontally on the surface of the soil, inside the soil, and it produces advantageous buds. If this is cut, the small portion can regenerate the entire plant. This is underground part. From these aerial parts are produced. Any mechanical force, any force is applied over that. This can be cut or detached, as a result of which, for example, a piece of ginger can be made into 5 pieces, and those 5 pieces can independently grow into an independent plant when provided the soil is good for its growth. So this is a kind of asexual reproduction. Then corm. See corm, colocasia, amorphopolis. Colocasia, amorphopolis are example for that. So this is also similar to that of the rhizome, but the only one thing is that this is growing horizontally. Rhizome is growing horizontally, but colocasia is growing vertically. This also produces axillary buds, and when a cut is made, this can grow independently to produce a new plant. And fourth is tuber, potato, Solanum tuberose. We know the structure of tuber. It is underground tuber, aerial part is there. There are certain eye-like spot. The eye-like spot represent the axillary bud. There is axillary bud in that. Each bud can grow into an aerial part and can produce a cluster of potatoes underground. And so, how many eye spots or how many eye spot represent the nod. In each nod there is a vegetative bud, axillary bud. Each axillary bud is capable of growing into a new plant. So, in potato, from a potato we can produce 10 or 15 potato, that will be depending upon the eye spot and axillary buds available. And bulb, onion, Allium cepa. So, we know bulb, the stem is much condensed. If you have the LS, you will see structure like this. This is the LS of onion. And this is the condensed stem. These are advantageous roots. And you can see axillary buds here. And these axillary buds, when it is cut, can grow into a new plant independently. In that way, as far as the stem is, the vegetative reproduction in stem is concerned, two categories of a stem: subaerial stems and its modifications, underground stem and its modifications are involving in the reproduction to propagate the own progeny. See, all the individuals produced because of that will be having the same genetical setup of the mother plant. So, in asexual reproduction, the progenies or the offsprings are similar to that of the mother. That is very, very important. The next is vegetative reproduction in leaf. So, vegetative reproduction in leaf. How is a vegetative reproduction possible with the help of a leaf? So, any advantageous bud, we have already talked, any advantageous buds or special buds produced from the leaf or stem or root can propagate. Vegetative advantageous bud produced in leaf like bryophyllum, scilla, and some other plants like this, they produce certain advantages. But say we take for example bryophyllum. Bryophyllum is a succulent leaf with side notches like this. See, in the notches it produces, there are advantageous buds. Each advantageous bud produces an young plant, and if it is detached from the mother leaf, this can grow into new bryophyllum. So, the succulent leaves are meant for storage of food and also to reduce the transpiration. But in the special category like this, the succulent leaves involve in vegetative propagation by producing vegetative buds and their notches. And there is another special plant called scilla. It is a bulbous plant whose growing in a sandy places, whose leaves are growing bulbously, long, elongated, like this, cylindrical leaves, at the tip of which they produce certain vegetative buds, which when they are detached from that, they fall on the ground and grow into new plants. So, in this way, leaves are also involving in vegetative reproduction, and they produce asexually. Vegetative reproduction is a kind of asexual reproduction whereby the new individuals produced are similar to that of the parent plant or mother plant. So, we have seen two examples in this. One is bryophyllum, another one is scilla. Advantages of vegetative reproduction. What are the advantages of vegetative reproduction? Number one, commercially important part. Important parts can be propagated quickly. Number two, these plants are genetically uniform. It can be produced in any season. Any season. And fourth point, farmers use this method of propagation to produce the plants, commercial plant in large quantity. So these are said to be the advantages of vegetative reproduction. See, vegetative reproduction, we know that the plants which are commercially important or produce quickly, and they are all genetically uniform, and it can be produced in any season, and farmers produce the commercial plant in large quantity by using these methods, any of these methods. And what are the disadvantages? Disadvantage. See, when we talk about the commercially important plant in large quantity, see, we should also remember about the plant hypomia. Hypomia batata, sweet potato. So that produces, and also dahlia. These roots are storing roots. They produce clusters of roots in the vegetative reproduction in roots. We can see each root produces an advantageous bud at the tip, and each tuber, this is a tuber, each tuber can grow into a new plant. So we should remember, farmers use large quantities. From one plant, many plants can be produced in this way, large quantities, using this method. And the only disadvantage in this, genetical modifications are not found. So genetical changes, as the organs are involving in the propagation, there is no chance for gametogenesis, fusion of gamete, and variation, etc. So there is no genetical modification, genetical variations in these type of plants. They are all the plants with the same genetical setup. So it's over. So in the vegetative reproduction, as we said, there are two types. One is natural method. We talked about the natural method in the earlier frame. And now the artificial methods. What are the differences between natural method and artificial method? Natural methods of vegetative reproduction happens naturally, the plant itself involved in that, and no external forces or no external agent involved in the agent is involving in this vegetative reproduction. But the artificial method is a method of reproduction in which external agents or the farmers or anybody is involving in that to make the reproduction. So that is called artificial method. The artificial method, there are two types of artificial method. One is conventional. To make it very clear, natural method, we have seen so many, ginger or any plants like that, the underground stem or aerial stem. When by accidentally the stem is cut, the axillary bud in that will grow into a new plant. So that happens naturally. Artificial measure, the agents are involving in that. For making the process, agents are involving, that is why it is called artificial method. In this there are two types. One is conventional method of asexual reproduction, another one is modern method. What is the meaning of the word conventional method? What is conventional? Conventional means traditional or old or something which is followed for a long time is called conventional method. Farmers have used many types of propagation of plant, many types of inventory methods to propagate plant. They are coming under the artificial methods and conventional methods. They make those methods to produce the new plants, produce those plants which are commercially useful, the plants which are having very important wanted characters to propagate. When they apply certain techniques over that, and this is used for a long time, that comes under conventional method. In the conventional method, we have method of vegetative propagation. The conventional method of grafting, we have three methods. One is cutting, second is grafting, third is layering. What is cutting? See, in cutting, cutting is a kind of vegetative reproduction by which either the part of the stem, root, leaf is cut into pieces, and each piece will grow into an individual plant. Each piece will grow into new plant. So for root cutting, apple, some part of the root can also propagate into a new plant. And stem cutting, moringa. So what is cutting? See, stem, it is cut this part or this part with the axillary bud can grow. This is commercially useful. Stem cutting is used in moringa and hibiscus and Boyer Hevia diffusa, bougainvillea, and in those plants stem cutting is used. So what is the meaning? Part of the stem, see, in villages you would have seen that people carrying the stick of drumstick, and to their places, when moisture is soil and provided water is the stick is watered, and that will grow into a new plant. So this is a stem cutting. In hibiscus also, the stem, part of the stem will grow into a new plant. A stick can be carried from one place to another place, and that will propagate into or grow into a new plant. And leaf cutting, as we have seen, bryophyllum, a part of the leaf bryophyllum, and is a best example for the plant that produces through the small pieces of leaf. So cutting is over. Next is grafting. So what is grafting? Grafting is a conventional technique which is used to fuse two plants and produce a hybrid. Fuse a stem like that, stem or buds one with another, and that will grow into a new plant. For grafting there are types. One is bud grafting, by which parts of two plants are brought together and they are fused and they will be allowed to grow into a plant. And there are types in the grafting. There are bud grafting. Second is tongue grafting. Third is crown grafting. Fourth is wedge grafting, and fifth is, we'll tell afterwards, bud grafting. So if a plant is taken, in its nodal region, an incision or cleft is made in the shape of T, the shape of T like this, and the bark is split open. Bark is split open like this, and a bud from other plant with an axillary bud with some other plant with the bark is brought here and placed, and the bark of the first plant is put in its original position, and polythene or tape is covered. After some time, the bud from the other plant will get a attachment with the early plant, and this is a kind of bud grafting. So here what is grafted? So attachment, what is attached? Bud is attached to the one plant, and we have two things in this we have to see, and the plant with, in these two plants are involving. The plant one is called stalk with root. The plant with root is called stalk. This is stalk. The another one which is used or whose part is taken is called scion. So two plants are involving in this, stalk and scion. Then tongue grafting. The fifth one is approached grafting. Two plants involving the tongue grafting also. One is called stalk, another is called scion. See, a plant with root is taken for experiment, and its trunk here, the plant 1 stalk and plant 2 scion should be in same thickness, and its stem is cut slantingly like this. And another plant, this is stalk. Another plant, that is scion, is also cut with leaf, is also cut in the same direction. This is brought here, and you can see when the stem of these two are brought together, and they will be joined like this. So the stalk of one plant and the scion of another plant, and see in this what is the necessity for bringing this. See, the scion have the aerial parts have are growing well, commercially important, and in the stalk root is growing well, that withstand the water scarcity, etc. So that this hybridization is a little somatic hybridization. It is a hybridization like process. So where two plants are brought together, and it is covered with tape or polythene papers like this. It is left for some time. There will be these two plants. After some time, these two plants will make the vascular connectivity among in between them, and it will grow into a single plant. Next is, we'll take approach grafting. So in approach grafting, so two plants are involving. One is bigger, it is having a branch like this. This is usually made in a mango tree. And another plant with which is growing in the pot is brought, and some portion of this is scion, upper plant, this is stalk. Both the stalk and scion, one side of the stem is removed, and here too the same way one side of the stem is removed. These two are brought together, and so ultimately they are brought together like this, and there is a off portion of the scion, off portion of the stalk or cut, and they are brought together with the help of a tape or polythene paper. It is glued and left for some time. What will happen? The vascular connectivity or vascular connection between these two plants will be established. Afterwards, the basal part of the scion and upper part of the stalk are cut and removed, detached. So the plant in the pot will be having its own basal part and upper part from the other plant. So in mango, this technique is adopted to produce hybrids. So this is called grafted varieties. This is resulting in approached grafting. And crown grafting. Crown grafting is a method where the stalk is having a bigger trunk and the scions are small. Stalk big, scion smaller. See, in the bark and the trunk of the stalk, some incisions are made or clips are made and they are opened. And the scions, the small plants, are brought together. Here they are placed like this. A connectivity is made using some artificial things. See, after some time there will be a connection between, there will be a vascularity connection between the scion and stalk, and this can be grown into a individual plant, or it can be left as such. The very near the ground it is detached, and new plants are growing over this. 1 or 2 or 3, 4, 5 bush like appearance will be the resultant of this grafting. Then wedge grafting. Wedge grafting, in the stalk, the stem of the stalk is broken at the center, and scion is taken. The scion, the both side it is cut and it is fixed. The scion is fixed in the stalk like this. So this is wedge shaped. The scion is wedge shaped, and if it is covered with some kind of polythene paper, etc., after some time the vascular connectivity, the xylem phloem connection will be established and the plant will grow naturally. So this is another kind of grafting. So we have discussed about 5 types of grafting. One is bud grafting. See, in all these cases 2 plants are involving. One is called stalk, another is called scion. The scion is the upper plant, the stalk is the plant with the root. And bud grafting, a bud is grafted to the stalk. And tongue grafting, the plant with the same size of the trunk are brought together, they are tied up. And in crown grafting, we would see the stalk is having bigger trunk and scions are smaller, and the many scions are made to attach with the stalk, a single stalk, and they can grow into a bushy structure. And wedge grafting, here the scion is the stalk is splitted into V shaped, in which the scion with wedge shaped cutting is inserted and a vascular connection is established. And as I said, the approached grafting, a plant, a potted plant is brought, and the stem of the potted plant is attached with the branch of the another rooted plant, and after some time hybrid is made. So these are the types of grafting. So in the conventional method of vegetative propagation, I was telling that there are 3 types. One is cutting, another one is grafting, third is layering. So we explained both, and now third type is layering. What is layering? Layering is a method by which the stem of one plant made to produce roots when they are intact or in connection with the mother plant. So this is called layering. In the layering, we have two types of layering. One is mound layering. This type of layering is adopted in Jasminum and Ixora. See, in Jasminum you would have seen that there are branches. If this is the soil, there are branches very near to the soil. If there are branches growing like this very near to the soil, what do? They bend the branch very near to the surface and keep it immersed or keep it bent, and they put and this is leafy shoot, this is soil, and the lower branch is bent and it is part of it is buried inside the soil. So if it is left within 1 to 4 weeks, you will see 1 to 4 weeks, the bent stem part of the stem part will produce the advantageous roots. So after some time, after 1 month, that is 4 weeks, this new plant can be detached from the mother plant, it can be grown newly. So this is a method of propagation. Actually, mount layering is a method of propagation. Here hybridization is not performed. This is mount layering. This is adopted in Jasminum and Ixora. Next is air layering. Second is air layering. If we want to make a new plant and from a particular plant, that plant does not have branches or branches very near to the surface of the soil, very near, and so what to do? So air layering is done. If the woody trunk with the branches are there, a branch is taken, and in that at the nodal region, the bark is removed like this, and the vascularity is there, and over that growth hormones are sprayed, and after spraying growth hormone, a soil sand with nutrients, and hormones, growth promoting hormones are applied. Clay and sand is applied over this place, and with the help of a polythene bag, it is covered and left for 2 to 4 months. See what happens, water supply is provided to this area, but the bark of this plant, the upper part, will start producing roots, and after 4 months this can be detached and this branch can be grown into a new plant. So this is a method. Air layering is useful for making a vegetative propagation in the woody plants whose trunks are or whose branches are very high above the soil level. But in mound layering is performed in the plants like Jasminum, where the lateral branches are very near to the soil. So layering is over. Now we will discuss about the advantages and disadvantages of conventional methods of propagation. See, advantages are all the plants produced by this conventional method are genetically uniform, and it is a quick method to propagation. It's a quick method. And third point, very, very important, some plants are producing poor seeds or no seeds. Which are commercially valuable, and the seeds are not viable. In those cases, vegetative parts can be used for the propagation through this method. And vegetative propagation of a Solanum tuberosum, plant like Solanum tuberosum, is possible because of this method. And the fifth point, when the two plant A and plant B are brought together and they are grown as a single plant, and we get the characteristic feature of both the plant in this. So these are the advantages of conventional method, but there are disadvantages also. So what are the disadvantages? There are only two. If virus infected part is utilized for this propagation conventional method, and again virus infected plants will be, the progenies will be produced, and there is no way for getting rid of or escaping from the viruses. That is one disadvantage. Second disadvantage is that it is the vegetative parts are bulky and it is very hard to handle. That is another inconvenience. And however, these when you consider the advantages, disadvantages are smaller and negligible. See, in the modern method of vegetative reproduction, we have an example. Professor F.C. Stewart of Cornell University isolated the phloem, matured phloem parenchyma, matured phloem parenchyma of the carrot, and grew it, the matured parenchyma, in a conditioned condition, grew it into a new carrot. So in the modern method, this is called tissue culture. So in tissue culture, there are two methods. One is totipotency. We will see what is tissue culture first. And micropropagation. What is tissue culture? Growing the cells, tissues or organs of a plant artificially in a conditioned environment is called tissue culture. So tissue culture is a modern method of propagation of plant in which the cells or tissues or organs of a particular plant is grown into a new plant, for which the concept of totipotency supports. What is totipotency? Every living cell is capable of having the inherent ability of producing the entire plant. Inherent ability of entire plant, and this is called totipotency. So a cell, a living cell, every living cell of every living plant cell, actually plant cell, is capable of producing the entire plant. This is called totipotency or total potency. And what is micropropagation? So micropropagation is actually a tissue culture method whereby cells, tissues are grown into the plantlets. Cells are grown into entire plant, and that is called entire plants, and this is called micropropagation. And when we study about the tissue culture, we will learn all these things in detail. See, in micropropagation, cells are taken, they are grown in a laboratory, callus is produced, from the callus small plantlets are produced. Those plants are similar to the mother plant but smaller in size, and afterward they are grown in the normal condition. From that many plants are produced. So in modern method is useful to produce plants in large quantity in a very short time. So modern method is useful for this kind of propagation. As a last segment of this session, we will see the advantages and disadvantages of modern method. See, advantages In a short duration we can produce the plant through tissue culture techniques, and all the plants are genetically similar and they are also called clones, and this can be performed in any season. So season restriction is not there. And some plants are producing very poor seeds and they are not viable, poorly viable seeds, but they are commercially valuable. To propagate the commercially valuable plants which are incapable of reproducing through the seeds can be propagated through this modern method. And there are certain plants which are endangered. The number is going on decreasing. So the endangered varieties, when naturally the number is going on increasing, to retain the plant on the globe, we take the leaf, stem, root or any part of the plant and we multiply them and grow them into entire plants. So in that way, lakhs and lakhs of plants from a single plant can be produced. So endangered plants can be preserved by this modern method. And virus meristem culture, taking a meristem, from meristem the plant tissue culture is adopted and we can produce virus-free plant. So virus-free plant is produced in banana through meristem culture. This is possible because of this method. And plants can be genetically modified by using this method. How? Plant A can be, cells of plant A can be inserted with a desirable gene and it becomes a recipient, and that cell can be multiplied and the cell will be having the qualities of two plants, both the plants. So genetical modifications can also be brought through by this method. So these are the advantages. And what are the disadvantages of this method? So this requires skilled laborers, and only skilled labor can involve in this. Common people cannot do this work, and so it is hard to perform. And the entire atmosphere should be kept in sterile condition. This is another threat to these modern techniques. Unless the entire laboratory or entire place is kept sterile, this the atmosphere will be spoiled. So this is also very hard. And the plants are having same genetical setups and same genes, and so they are susceptible to the germs. If one plant is, all the plants are, we said that they are all clones, they are all having same genetical setup. Since one plant is infected, all the entire population will be devastated. So that is another disadvantage. And last thing, see, callus undergo genetic disorder, as a result of which expected results may not come sometime. So these are the disadvantage, and this topic is over. We have, in this session, we have discussed about the method of asexual reproduction, not the sexual reproduction, asexual reproduction. In that, vegetative reproduction. The vegetative reproduction, what are the conventional method and what are modern method? Natural method, artificial method. In the artificial method, conventional method and modern methods have been discussed so far. And if you go through the books, you can get it clarified furthermore. Thank you.